If I can, I would like to maybe provide a little bit of background. There's a lot of schools of thought that kind of went into this, and I think that maybe providing a little bit of background on the study, and then how that evolved into what we did through the MS Pass program would probably be really interesting for your audience and your listeners.
Yeah, absolutely.
So what fueled this project specifically is that there are many different treatment options for relapsing forms of MS. And personalized predictions of what we call treatment effects have become really critical for clinical decision making, especially given the variable safety and efficacy profiles of these different disease modifying therapies for relapsing forms of MS. And patients often experience different outcomes under the same treatment. And the baseline risk of patients is often a determinant of what we call these heterogeneous treatment effects. So there are many patient characteristics, including age and disability status and disease activity and comorbidities, that can affect these overall baseline risks. So this work in particular was based off of published two-stage models of heterogeneous treatment effects that were previously derived from clinical trials that evaluated 3 different disease-modifying therapies: glutaminyl acetate, dimethyl fumarate, and natalizumab. And the authors used a risk modeling approach to predict the probability of patients having an MS relapse within 2 years, and those with different characteristics treated with any of those 3 different MS disease-modifying therapies. So the term two-stage came from the development of 2 different stages or phases of this predictive modeling to predict the risk of having an MS relapse. And this was the outcome measure that those authors decided to look into. So the first stage involved the development of a baseline risk score, which linked the probability of an outcome to patient characteristics that were felt to be important indicators for selecting a particular disease-modifying therapy. And then the second step then involved the estimation of the probability of post-treatment outcome as a function of that baseline risk score. So the authors were then able to apply those data to predict MS relapses in 2 years under different treatments using that individual patient data from clinical trials. So this work showed the potential for improving personalized medicine, though it had limited generalizability due to restrictive inclusion criteria that aren't inherent aspects of randomized controlled clinical trials. So bringing it back a little bit to what we were particularly interested in is the notion that generating models from real-world MS data can have additional complexities. This can include broad demographics and MS phenotypes, non-standardized visits, unequal treatment distributions, and then lack of randomization or placebo control. However, real-world data-based heterogeneous treatment effect studies have the additional advantage of generalizability relative to randomized controlled clinical trials that provides an excellent substrate for this risk prediction. So using that data, our overall objective was therefore to establish a proof of concept for this 2-stage heterogeneous treatment effect model, specifically for relapse, that was inclusive of all disease-modifying therapies and MS patient types using real-world data, and compare them with the published clinical trial models that I just summarized. So using the MS Pass framework, we then conducted our own heterogeneous treatment modeling study that included patients with relapsing or progressive MS who started a disease-modifying therapy with at least 1 year follow-up without a change in disease-modifying therapy who had a complete baseline data set. And then the disease-modifying therapies were then grouped into broad efficacy categories of high, moderate, and low effectiveness. And so patients were then split into training and testing sets that were stratified by these treatment groups. And we then conducted the first step to analyze the performance of the overall model to make sure that it essentially worked. And in the second stage, we then derived a propensity score model to balance our baseline covariates, because as opposed to the work that was done with the randomized clinical trials, these data were based on real-world populations, so we needed to correct for indication bias. So that was an additional step that we had to take in our model. We then use low efficacy disease-modifying therapy as our reference point. And combining these two steps, baseline risk modeling and then the propensity score modeling, we were then able to conduct our treatment effect modeling.